at your own school? Because I know that a lot of you have told me, like, for the previous lessons we've had, you guys learned a lot of that. You've covered a lot of related stuff at school. So I think the today's topic should be pretty new to you guys for the most part. So first, we're just gonna do some icebreakers. First, who is your favorite fictional character? It can be from like a book, movie, show, video game, pretty much anything. So we're just gonna tackle that one first. Just put in the chat. Uh, and yeah, we'll see what everybody says. Okay, well, we're waiting for people to put their ideas in the chat. I guess I'll tell you guys mine. So like in middle school and stuff, I really enjoyed reading Rick Riordan books. So my favorite fictional character is Percy Jackson. Kind of kind of a classic. Oh, Anna says, I don't know. That's fine. Okay, so if you guys aren't really sure about that one, why don't you just say, what do you think you might want to be when you grow up? It doesn't have to be for sure, but just just put an idea in the chat. I would say, for me, something in the medical field. Any ideas? Just type it in the chat. There's no wrong answers, so just putting ideas out there. Okay, well, I'm just gonna move on then. So, we're almost done with the course actually. This is week 5. We only have next week left. So
What did we do last week? I wasn't there.
last week, we covered mutations and genetic disorders, genetic variation, but we focused on
Yeah, last week
mutations.
for some reason the screen just kept saying host has joined. Let them know you're there, and then for about like 20 minutes or something, nothing happened.
Oh, really? Did you try joining? Was that when you tried joining the Zoom?
Yeah.
Oh yeah, I think yeah, hopefully that won't happen again. Sorry about that. But yeah, that's not something I can control. Any more questions? Okay, great. So this week we're doing genomics and genome annotation. That's just the basic gist of it. We're gonna go more in depth um with each of these topics. And yeah, last week we did mutations and genetic disorders. I hope you guys got a chance to do like the homework, which is basically just to write a paragraph about a genetic disorder that you picked. Do some research, which I thought would be an interesting thing for you guys to do. So yeah, let's get started. First, we're gonna just do a review of transcription and translation from last week. I know we watched a video on it, but I'm not sure if you guys got the basic gist of it, so we're just gonna go over it real quick. Then we're gonna go over genomics, genome annotation, and then we're also gonna go over some ethics and more stuff. So yeah, a lot to cover. So first, we're gonna do a review. Basically, transcription. Let's just say, imagine your DNA is like a giant recipe book, right? We've used these kind of analogies before, during the first few weeks of the course. So we already know what our DNA is. It's like the set of instructions that builds and makes up our body, makes us who we are. It's pretty much the basis of all genetic information. So yeah, so imagine your DNA is like a giant recipe book in the library of your cell. So when your body needs to make something like a protein, it can't carry the whole recipe book to the kitchen. So instead, it just makes a copy of that recipe that it needs. So that's kind of like that's just an analogy that might make it easier to understand what transcription is. So it's basically like making a photocopy of the recipe from the book. Um, and in your cells, there are special machines that read the DNA and then make a copy of it. So this copy is actually translated into, or not translated, it's made into messenger RNA, which is mRNA. So then it's kind of portable and you can quote unquote leave the library and go to the kitchen. So it's basically just a way to transport copies of things, or just make a copy and so then you can later transport it. And that goes into translation. So your mRNA, which is the portable recipe, the copy that you made, it goes to the kitchen of the cell called the ribosome. So basically, the ribosome is what makes proteins. So yeah, this mRNA is needed to make proteins from the DNA. So the mRNA is read by the ribosome and it tells the workers um which order to create the specific protein. So basically, it carries all the instructions carries all the instructions to make the protein, basically. So yeah, each set of um three letters in the mRNA, it's called a codon. And we'll go over that more later. It represents an amino acid. The workers bring these amino acids and they link together in the order specified by the mRNA recipe. So when all the amino acids are lined up correctly, that makes a protein. It's also called a polypeptide, which is just a fancy word for protein. Um, so yeah, they're really essential because they do a lot of different jobs in your body, like helping your muscles move, carrying messages between cells, and digesting food. So yeah, that can be confusing a little bit. So any questions on that? Transcription or translation? So transcription is basically just making a um, copy kind of, and then translation is when it's actually translated into the protein. Does that make sense? Any questions? Okay, I'm gonna move on. So basically, just in summary, like I just said, transcription is like copying a recipe from a cookbook, and translation is like following that recipe to cook something delicious. In this case, a protein. So yeah, these two processes work together. They're really intertwined. Okay, and here's just a diagram to help show you what it's like. You see, the first one is basically a DNA. You see the strand, you see A paired with T, C paired with G. Those are the bases. Remember we went over that with DNA structure. So yeah, we see the first image is normal DNA, right? Then in transcription, it gets translated to RNA. Basically, in RNA, instead of thymine, which is the base, which which is what T stands for, we have uracil, which is what U stands for. So A pairs with U in this case, um, and C pairs with G like normal. But yeah, that's really the one of the main differences between RNA and DNA. So then after RNA is translated into or transcribed into or DNA is transcribed into RNA, then we have translation, which basically uses the RNA to create a polypeptide. So each codon is changed into different amino acids. Any questions on that? It can be a little confusing since we haven't gotten the chance to go more in depth on how this works exactly, but this is the basic gist of it. It's kind of good to know. But yeah, we're gonna be focusing more on the genetics of part of this. Don't be afraid to ask questions if you don't understand anything. Okay, so here's a big picture. What is genomics? Basically, genomics is the study of all the genes in an organism and also how they interact with each other and the environment. So another analogy is like reading a book, but instead of words, we're reading genes. So it's important to know the difference between genetics and genomics. Um, genomics is more of like a holistic approach. So it's just the study of all the genes, um, and all the genetic information in your body and how they interact. What is a genome? A genome is the complete set of an organism's genes. So another analogy is like a giant instruction manual that guides how an organism grows, functions, develops. So it's a complete set of an organism's genes. It's just the entire thing, the genome. Got it? Okay, so why is it important to study genomics? Like, aren't we already doing this with genetics and stuff? But understanding genomics is really important for, biomedical research. We can understand diseases and how to prevent them and treat them. And also personalized medicine, which I think I talked about a little bit before, which is basically tailoring specifically to an individual's genetic makeup to see how they react to certain treatment, to make it personalized towards them. And evolutionary studies. So yeah, it's really important for genetic disorders like cancer genetics and the identification of diseases, and also the mutations that cause them. So first we have structural genomics. Basically, structural genomics it's a type of genomics, and it aims to determine the structure of every protein encoded by the genome. So it's basically just focusing on protein structure, the proteins that are made by the genome. So yeah, it's kind of self-explanatory since structural is in the name. So that won't be too hard to remember, hopefully. We have functional genomics. This aims to collect and use the data from sequencing for describing gene and protein functions. So similar to structural genomics in the way that functional genomics focuses on function, while structural genomics focuses on structure, right? So functional genomics explores gene functions, interactions, and networks. So techniques like CRISPR, which we talked about, I think last week, with the gene editing and stuff, it helps researchers study gene functions by altering specific genes and, studying the effects they have and how it can impact the body. Then we have comparative genomics. So basically, it's involving comparing the genomes of different species to understand, the evolutionary relationships and identify conserved elements. So yeah, comparative genomics is based on comparing things, comparing the genomes that different species have. Like, why are they different? Like, what in evolution caused this change, or why is this way? You know? Um, Oh yeah, also another term you might want to know, orthologous genes. They're basically genes in different species that evolved from a common ancestral gene. Genomic medicine. So basically, this uses genetic information to guide medical decisions, diagnosis, and treatment. So it's kind of like uh, similar to personalized medicine. And we also have pharmacogenetics, which is the study of how genetic variations influence response to drugs, allowing personalized drug prescriptions. So yeah, this really ties into personalized medicine because if you didn't understand already, pharmacogenetics, genomics, is basically, running tests on drugs and treatments, and seeing how certain groups and their genetic makeup you know, impact their response to the drug. So maybe one person with one genetic makeup or different genes, they might have a different response to the drug than another group of people with different genes. So genes can impact people's response to treatment. So studying pharmacogenetics can allow it. Genomics, yeah, usually they're kind of interchangeable, but pharmacogenetic genomics is more about looking at the bigger picture. But yeah, so it allows us to, see what treatments work better for which group of people with certain genes. So yeah, kind of like personalized medicine. Here's some areas of research. There is epigenomics. So this can kind of be a confusing concept. It's basically the study of changes in gene function that don't involve changes to the underlying DNA sequence. So that includes DNA methylation and histone modification patterns. We're not going to go over those too deeply because, we could spend hours talking about those two things. But if you want to, you can research into those on your own time. But yeah, basically they influence gene expression and are really important for um, development and also diseases. So yeah, that might be a little confusing, but that's what epigenomics is. Then we also have metagenomics, which is basically analyzing genetic material recovered directly from environmental samples. It's used to study microbial communities in soil, water, and the human body. And it's really important for helping scientists understand the diversity that this planet has and the applications that there might be for biotechnology. And we also have a really important part of genomics, which is cancer genomics. Genomics, obviously, cancer has been it's like a really big object of discussion and study. Um, scientists are constantly trying to find a cure for cancer. Treatments that might help it make help people cope with it better. So it's basically understanding the genetic basis of cancer. So that includes identifying oncogenes. And oncogenes are basically the genes that cause cancer. And also tumor suppressor genes. Tumor suppressor genes are a really interesting concept. I might be able to talk about it a little bit. Oh, Anna said my dad treats cancer. That's really cool, Anna. Yeah, great. It's definitely a hard concept to grasp and make more discoveries about. But yeah, scientists are trying to learn new things um all the time. So yeah. Um so yeah, it's kind of where was I? Yeah, so genomic analysis helps in personalized cancer treatments and early detection methods. And we're not going to go too much into how cancer works, although we could, but I might provide you guys with a video, or you guys can do your own research because it is a really interesting topic to learn about. But yeah, and also I did do some studying on tumor suppressor genes. Um, I think it was also a really interesting topic for me. So you might want to check that out as well if you're up for it. Any other questions? Yeah, anyway, if you wanted to learn more about tumor suppressor genes, basic gist of it is that they're genes that are meant to, suppress tumors and the kind of things that cause cancer. But if there's a mutation in the tumor suppressor genes, then that allows cells to multiply, um, and multiply without regulation, which can lead to cancer. So now let's move on to genome annotation, decoding the genome. So basically, this annotation is the process of identifying genes and other features in a genome. So it's basically a more formal way to put it is saying it's a process of deriving the structural and functional info from a protein or gene from a raw set of data using different analysis, comparison, estimation, precision, and other mining techniques. So, it's a really complicated process, but um, it's really just an important part of trying to understand the genome. really important for research. So gene identification. So scientists use a lot of different tools to try to identify these genes. They can do things like looking for specific patterns in the DNA sequence to pinpoint where the genes are located. Um, specifically to what they're studying. Let's go over some types of genes. We have non-coding genes and coding genes. So coding genes basically provide instructions for making functional proteins, which are really important for cell structure and function. On the other hand, non-coding genes, this can be like microRNAs and long non-coding RNAs, which you might not be familiar with. Basically they regulate gene expression and they play vital roles in cellular processes. So areas of research in genomic annotation, we can talk about alternative splicing. So basically, this is the way that many genes can produce like different forms of messenger RNA. So it's really important to understand this because it expands the potential diversity of proteins that can be that can be made from a limited number of genes. And also for non-coding RNAs, um, they're really important in gene regulation development and diseases, like we talked about on the last slide. We also have comparative genomic annotation. So we kind of already touched on this with comparative genomics, but specifically for comparative genomic annotation, it's comparing annotated genomes of different species to identify conserved genes, regulatory elements, and evolutionary adaptations. So it's pretty much the same thing, and it helps us understand evolutionary processes and the functions within the genome. And then we have genomic variation, which we've already covered a lot over this course. Um, so it's like studying genetic variations like single nucleotide polymorphism. Sorry, polymorphisms. It's a mouthful. They're called SNPs. Also, they're really interesting. Might want to research them if you're really interested in genetics. Um, so yeah, and copy number variations among individuals and populations. So yeah, these are also really important for personalized medicine. Any questions? We also have integrative genomics. This is basically combining genomic data with other biological data like proteomics, metabolomics. Those are really probably terms that you're not familiar with either, but we obviously can't go over all of them because, we definitely don't have enough time for this course. But I really suggest that if you're interested, you can go research these if you'd like. So basically, yeah, it's to understand um the biologic processes and systems as a whole. So it's more comprehensive because you're identifying and combining like a bunch of different data points. So there's also genomic databases. I've used a few, um, also kind of interesting to explore. There are a lot of publicly available ones like GenBank, Ensemble, NCBI's Genome Data Viewer, which I've actually used before. So yeah, they're really interesting, especially when you know what gene you're studying. Or like you're studying a specific gene and it gives you like a whole bunch of different data to use in your research. So now we're going to move on to ethical and social implications. So obviously with all this data um and information about genetics and DNA sequences, um it raises a lot of ethical concerns about privacy, consent, and discrimination involving genetics. So bioethics committees and regulations help address these issues by ensuring the responsible use of this genomic information, but there's still a lot of controversy about it, and a lot of thought that needs to go into, you know, these rules and, how scientists are supposed to deal with this information. So I'm just going to go over, some of the main ethical concerns that there have been with this topic. So first, as I already mentioned, we have genetic privacy. Individuals can worry about the privacy of their genetic information, and making sure that it's secure and not misused. We also have informed consent. Obtaining informed consent from individuals for genetic testing and research is really necessary because you can't get people's information without consent, and people have to understand, the consequences or implications of sharing their genetic information. And then we also have genetic discrimination. These are um, concerns about genetic discrimination in employment or insurance based on genetic predispositions or conditions. And legislation is needed to protect individuals from this discrimination. And also ownership of genetic information. So it's kind of complicated to determine who actually owns genetic data, especially with research. Um, so yeah, clear guidelines are needed to address these ownership problems. Then we also have ethics with gene editing. So, this is definitely also a heated topic because, there are a lot of ethical implications with technologies like CRISPR because it raises questions about, should we be modifying human genes like this? Like how can it affect future generations, right? Because it's a big deal, gene editing. But it's also a really important step for the future of genetics. There's also access to genetic services. Disparities in access to genetic testing and therapies based on socioeconomic factors can lead to ethical concerns because ensuring, equal access is really important as well. And then the last bullet point, kind of put it there for fun. There's also this, you guys heard of designer babies? So it's kind of like the concept of like giving um unborn children like specific traits. Like, parents would be able to pick out the specific traits that their children would have. So it wouldn't be based on chance anymore. So this is definitely like a an ethical problem because if it kind of, raises questions about the limits of genetic engineering and the definition of normal, so to speak. So yeah, we can't let genetic um editing technologies to get out of hand. So there always has to be, rules, and regulations, committees on ethics that look out for these kind of things. We also have future trends. So advances in single cell genomics enable studying individuals' cells genomic profiles, revolutionizing our understanding of cell diversity. And precision medicine continues to evolve, which offers, tailored treatment based on the patient's genetic makeup. So why genome annotation? Why is this so important? Kind of already covered this a lot already, but here's just a little nice sum of it. So it basically helps us understand genetic diseases, evolution, and even design genetically modified organisms like GMOs for agriculture. I'm not sure if you've heard of those before. Um, but yeah, it's like having a map to explore the genetic world, basically. We can't study the genome without identifying all these different specific parts, because then we can identify how they interact with each other. So yeah, you see this picture on the left looks pretty complicated. So these are just some of the different ways to track genetic data to see the big picture. So yeah, these can be really confusing looking, but this is an example of a circos plot. Um, I've never had to read one before, but But yeah, this is what some researchers use to help make scientific discoveries. So yeah, so it'd be interesting to see that graph kind of because it's has a lot of obviously has a lot of different components. It shows how complicated genetics can be, even when you're only focusing on one factor of it. So applications of genomics. Genomics has a lot of real world applications. We've already talked about precision medicine, you know, developing personalized medicine, to tailor people's genetic makeup to actual individuals' genetic makeup. Then we have GMOs, which I just mentioned not that long ago. Like creating genetically modified crops that can be more resistant to diseases and pests, in the environment that they're in. Then we also have conservation, which is studying and conserving species that are in danger of extinction. Okay, here's just a practice exercise. Um, what we're basically gonna do is you're gonna try to fill in the terms. So there are some, see some of the terms on the bottom. Just try to fit them in into the right boxes. So let's just go over this. First we have DNA, that was the example. Those are molecules containing genetic information. Example of that, inherited genetic material. So yeah, let's just try to fill in each box. This is kind of review. I just want to see how much you remember from, the first few weeks. Um, so yeah, this is pretty much review. What do you think goes in the second box? The box right under DNA. Any guesses? Structure housing multiple genes. Humans have a total of 46. Either say it out loud or put it in the chat. Look at the bottom for the choices. Come on, guys, try it out. Don't worry if you get it wrong. Yeah, Bryce said chromosomes. That's right. Great job, Bryce. Thanks. So yeah, we have chromosomes right there. We got one down. What about variant of a gene that can influence a trait? And the answer is kind of obvious because the example says allele for hair color. So we already know it's what is it? It was kind of an example already. Okay, well since nobody's put in the chat, it's an allele. It's basically just a different form of a gene that determines someone's traits. So what about a change in the DNA sequence? We covered this last week. And the examples are pretty much giving it away, but mutation causing albinism. So yeah, it's a mutation. Transmission of genes from parents to offspring. An example of that can be blood type heredity. It's kind of funny because all the answers are already in the examples. I just wanted to review these. Yeah, Bryce said heredity. Great. Yeah, and then the last one, set of genes of an individual, that is genotype. So yeah, that's just some review. Just wanted to see if you guys remembered what you've got. So there are the answers. Here is another exercise activity. Do you guys want to see if you can fill in the blanks? What do you guys think? How about how about you try any of them? If you can do whichever number you want. Just in the chat, type number blank and then write your answer. Like if you say number four, you'd be like number four, and then you type in your answer. So you can choose any one of these and type in your answer. Which one do you know? And just type in the chat or say it out loud. Or number three. Three might be an easy one because we could just cover that on the last slide. Cover that on the last slide. Any ideas? Okay, let's tackle three together. Number three, humans have a total of 46 blank, which contain thousands of genes. You guys forget already? Chromosomes. 46 chromosomes. Okay, let's see number five. A mutation is a change in the DNA sequence, which can result in new traits or heredity disorders. Number six, heredity is the transmission um, of traits from parents to offspring, contributing to, or genes, or maybe it's alleles from parents to offspring, contributing to the variability of traits. Okay, well, here are the answers. I'll let you guys look at it for a little bit since it seemed like you guys might have had trouble with it. Any questions? Okay, here's another one. How about we only do number one and two, the first one and the bottom one? Because other than that, it's review. So what is the genome? Do you guys think it's A, B, or C? A is a type of cell in the body. B is a segment of DNA carrying instructions for a specific trait. And C is the complete set of genetic information in an organism. What do you think it is? A, B, or C? No wrong answers. Just put a guess in the chat, please. What do you guys think? Do you guys remember? Were you listening? Okay, we're gonna move on soon. So just type in whichever letter you think might be your best guess, and just put it in the chat. I want all of you to try to do that. Okay. What is the molecule containing genetic information? Is it A, RNA, B, provide instructions? Actually, this question is worded wrong. Um, the actual question should be, what are non-coding genes? Yeah, it must be a wrong or a wrong question. Okay, for what is a genome? The answer was the complete set of genetic information in an organism. What are coding genes? It's provide instructions through genes that provide instructions for making functional proteins essential for cell structure and function. So for conclusion, genomics and genome annotation are the forefront of scientific discovery. They shape the future of medicine, biology, and biotechnology. So by decoding the genome, scientists are figuring out new solutions to complex health challenges, like genetic disorders and cancer, and advancing our knowledge of life. Okay, great. So why don't we play quizzes? I'll stop sharing for a little bit and I'll share my screen. Hey guys, please join the game.